So when we think about phospholipids, these are amphipathic molecules. And amphipathic means that they have a polar head and a non-polar tail. Okay. So this gives these molecules this sort of polar property where part of the molecule can interact with water, which is the head, hence the hydrophilic, right? It's going to um, be loving water. Philic comes from affiliation or a connection to. So hydrophilic means that this part of the molecule can interact in aqueous environments or can interact with water. Whereas the tails, the nonpolar tails, these do not interact very nicely with water. So this is what we mean by being amphipathic. It has properties of polar and nonpolar molecules as well. And that becomes really important when we think about several structures in the body. One is the phospholipid bilayer. This is the membrane that is encapsulating all of our cells. This forms the cell membrane. And so if you think about the nature of these molecules, right, the head can touch the water environments, which would be inside the cell and outside the cell. But then the tails sort of face each other and create this barrier, which is perfect to form a membrane. Another important thing we want to recognize with the phospholipid bilayer is that non-polar things. So, for example, molecules that are more fatty or fat affiliated can go through the phospholipid bilayer because they can exist in the center, whereas polar molecules cannot go through this phospholipid bilayer. So the membrane is not only good at separating two environments, but it's also this sort of selective barrier as well. Some things can get through there and other things cannot. Another example where we see phospholipids in the body is the micelle. And again, because our bodies is made of so much water, micelles help us move fats around. So when we think about moving triglycerides around from where they are absorbed in the gut to get them into the liver where they can be processed and stored, they need to move around in water. But by nature, they do not interact very well with water. So this little micelle is kind of like this protective bubble, if you will, that encapsulates the fats. The fats will exist in the center of the micelle, and then the polar heads can sort of move this around in the aqueous environments and the different water compartments in the body. So micelles are important transport mechanisms for fats inside our body where there's so much water. A third example of where we see phospholipids in the body is surfactant. And surfactant is an example of a phospholipid that's made in the lungs, and it helps to prevent the alveoli from collapsing. It's really important when we think about respiratory disease in infants. So infants that are born, especially if they're born prematurely, they don't have adequate amounts of surfactant. And so their alveoli collapse pretty easily. And this usually leads to infant respiratory distress or infant death. So surfactant is another really important clinical example of where phospholipids are and how their functions, them being amphipathic, is important to different regions in the body. So phospholipids, such as surfactant, keep the alveoli open by interacting with the water, but also sort of breaking up those interactions so that the alveoli can stay open. Okay, so 3 examples of phospholipids. These are one definitely to remember or commit to memory or star, just because they're so important clinically and to our function. Let's move on to eicosanoids. And eicosanoids are modified fatty acids. Essentially, we take a fatty acid tail or fatty acid chain, and by interaction with different enzymes, that fatty acid can sort of start to fold upon itself to form this much more complex eicosanoid. So we typically derive it from arachidonic acid, which is that really long fatty acid chain, about 20 carbons or so in there, and then we fold it on itself to make a more complex molecule. Examples of this are prostaglandins and thromboxanes. Now, the functions of eicosanoids generally, but more specifically, these 2 examples, is signaling molecules. They're really important for communication and sending signals. Thromboxanes, more specifically, are going to be important for blood clotting. So if you think back to that example I gave about positive uh, feedback, which was where clotting began at the site of an injury of a blood vessel, and we had more bleeding in order to deposit these clotting factors at the site and to help form that clot that can stop the bleeding. So thromboxanes is one of the first molecules that arrives at the site of that clot, and it pretty much sends the signal like, hey, everyone, there's an injury here. You guys need to start laying clotting factors here. So eicosanoids are going to be important for communication and sending those types of signals from one cell to the other. All right. And if you look at their structure down here, that would kind of make sense. They sort of latch onto the site, and then they act as sort of a flag that's being waved to signal other molecules. Let's move on to steroids. And steroids are another very important class of lipids. These are derived from cholesterol wings. You can see these much more complex carbon wing molecules. And the examples in the body are estrogen, testosterone, aldosterone, thyroid hormone, cortisol. Lots of different hormones in the body are going to be derived from cholesterol or steroids, okay? This is why steroids are so important, especially in the developing fetus, but more so in pregnant women. Like hormones in general are really important for these women. And then this is also why fats are important in hormone production. You've got to have a lot of healthy fats in order to be able to build these steroid rings. Okay. Let's shift gears and think about proteins. This is our third class of biomolecules. These are also essentially monomers that are attached to each other to form more complex structures, very similar to what we saw with the carbohydrates. So proteins are polymers of individual amino acids. So these are the singular or structural units that form proteins. And so amino acids can be different types. There's about 20 different types of amino acids. We can also classify them on the basis of whether they are made in the body or not. So we can think about essential amino acids, which are not made in the body and need to be supplemented through food, versus non-essential amino acids, which can be made by the body. Okay, and these are going to be the building blocks that help us form proteins. Now, there are peptide bonds, which are slightly smaller chains of amino acids or slightly shorter proteins. And so the way that we form them, again, very similar to what we saw with carbohydrates, the covalent binding, which releases water, hence this is a condensation reaction, and then forms this peptide bond. So these two amino acids joined together is going to be an example of a peptide. So a peptide is a shorter chain, about 2 to 50 amino acids covalently bound, versus a protein. Anywhere beyond 50 amino acids bound together is where we get into the realm of it being a more complex protein. Okay. Now, on that basis, we can think about different levels of protein structure. So proteins are crucial in many, many aspects of our physiology. Proteins are enzymes. Proteins are hormones. Proteins can be bound to other molecules such as glycoproteins, which are important for, again, cell signaling and cell recognition, or lipoproteins, if we bind a lipid to a protein, and that's going to be important for transporting that lipid around. So proteins can be found in several different regions of the body, and they have a lot of different functions, right? They also receptors that help for binding of cell surface, one cell surface to the other as well. Now, the levels of protein structure are going to determine the complexity of the protein. So we start with the basic level, which is the peptide bond formation. So we form this peptide bond, again, one amino acid to the other, and then we form a peptide, and then we form a more complex protein. So this is the basic structure or the primary level of the protein formation. Next, we have the secondary structure. And the secondary structure is when we have one amino group binding to the carboxyl group on another part of the very same protein chain. So this begins to form slightly more complex folding of our initial protein chain, right? The amino group here binding to the carboxyl group, and we can form two general types of secondary structures, the alpha helix. So this molecule can begin to take this helical turn, this helical formation, almost like a strand of hair that's curled. And then the second one is the beta pleated sheath. And it's the same formation of the amino group to the carboxyl group, but this takes a different sort of configuration where it is a loop, kind of like a hairpin, but then it's pleated along the length of that loop. Okay, so the beta pleated sheath and the alpha helix. So this is the secondary level of folding, and notice that this is beyond the initial primary level, which is just that peptide chain. Let's look at the tertiary level. So yet another level of complexity. This is when we have several different secondary levels or secondary proteins coming together to form a much more complex structure. So in this protein, we have some typical peptide bonds or protein chains in the purple. We have an alpha helix also sort of bound in there in the blue, this darker blue, and then we have a beta pleated sheath that is also bound in the lighter blue. So this is one general molecule. It's much more complex. This would be more like a receptor or maybe an enzyme of some sort. And its function is going to be determined by how stable these bonds are. So the initial bonds that we form, the peptide bonds that make this a chain, those are pretty stable, right? Those are covalent bonds, very stable. But as we start to get into this more complex configuration at the tertiary level, these bonds are less stable. So these can be interrupted by heat, by excess acidity, right? If the pH gets very low, or even just by mechanical disruption of these bonds. So what I say that to say is that as the structure gets more complex, it gets a little bit less stable as a trade-off. Lastly, we have the quaternary structure, and here we can see four completely separate proteins that are coming together to form one very complex molecule. So this would be a tertiary structure, this would be a tertiary structure, and we have four of those coming together to form an even more complex molecule. Now, this is an example of a protein that's found in our body. And as we can see, it has these other sites internally where we can bind other molecules. So this is going to be a transporter, very important transporter. And I want you guys to take a guess as to what you think this protein is. It has sites to bind four of something, I'm not going to say what. And then it has these four sort of specific tertiary proteins. So what do we think this is, just based on that description? Feel free to drop that in the chat. What do we think this is? I'm hearing red blood cells. That's a really good guess to start, but not quite. It is related to blood and the function of blood. It's actually found in a blood cell. Really good. Any other guesses on this one? Yes, exactly. Perfect. Hemoglobin. So this is hemoglobin. This is the structure of hemoglobin, right? I'm seeing multiple responses for that. And you can see the sites where heme would bind. So this is the heme part of this molecule where we would bind iron. And then we have the globin part, which is the protein, right? The much more complex quaternary structure of the protein. Okay. All right. Let us look at nucleic acids. Nucleic acids are the fourth and final group of biomolecules, and these are essentially our building blocks for the genetic material. So this is DNA and RNA as well, right? The building blocks of our genetic material. Now, we want to think about the structure of these nucleic acids as being pretty standard, but there can be some variation. So the standard formation is a phosphate group, which is here on the left, a 5-carbon ring, which is in the middle, and this can differ. If it's a deoxyribose ring, it's going to have, which means it has one hydroxyl group and one hydrogen. This is a deoxyribose. This is going to be DNA, right? Deoxyribonucleic acids versus if it only has, or if it has two hydroxyl groups, it's going to be a ribose ring instead of a deoxyribose. So this can be DNA or RNA. And then the third component of this molecule would be the nitrogenous base. And the nitrogenous base can also have some variation, which would further classify types of nucleic acids. So if it is a pyrimidine, that general class, it's going to include nitrogenous bases that are cytosine, thymine, and uracil. Whereas if it is a purine, it's going to have nitrogenous bases which are double ringed, and they will be adenine and guanine, right? So depending on the base that is here, this will determine what type of nitrogenous base and then what class, whether it's a pyrimidine or a purine. Let's talk a little about the different functions of these nucleic acids. So we have different types, but their general function is to be energy transferring molecules. They help us to move energy around in the body. And so one of the examples is AMP, which is adenosine monophosphate, diphosphate, or triphosphate, depending on how many phosphates are attached. And when we talk about cellular respiration, we'll talk about the way that phosphates are used as energy storage. And so if we have ATP, which is 3 phosphates, then we have the ability to cleave that third phosphate and to liberate that energy in order to do work, right? In order to do cellular work. So this is how we can shuttle energy around. And one of the major roles of the food that we eat to nourish our body is to make ATP. ATP is our biggest energy currency, and it will help us generate the energy that's needed for all of what the body is able to do. Another example is NAD, nicotinamide adenine dinucleotide. And NAD is another type of energy transferring molecule, but this one is going to transfer electrons, right? Electrons. So we can have NAD in the reduced form, which is no electrons being carried, versus NAD in the oxidized form, which is when, reduced form is going to be having electrons, and oxidized form is going to be no electrons, right? The oil rig, if you remember that mnemonic. Oxidation is loss of electrons, reduction is gain of electrons. And so both forms of NAD will be seen when we talk about cellular respiration as a way to move electrons around, which is another form of energy transfers. Same thing with FAD, flavin, adenine dinucleotide. This is also going to be FAD plus or FADH2, which is in the reduced form or oxidized form, and it's another mechanism of moving electrons around. Okay. Another type of nucleotide are cyclic nucleotides. These can form rings instead of phosphate chains, as we just saw. And so this is going to form cyclic AMP and cyclic GMP. These are also messenger molecules that are used in many of our signaling cascade processes, the way that we allow cells to, especially with receptor binding, cells can receive instructions on internal processes by way of the signaling cascade or the signal transduction. And so cyclic AMP and cyclic GMP are molecules that help to pass those messages along, okay? So when a receptor binds, the receptor tells another molecule to do something else, and that molecule tells something to do something else. And so cyclic AMP is a way to pass that message internally to bring about the cellular function.